Wow, it's so awesome to be on this stage. Good morning, everyone. So we're Team Plasticity from Imperial College, London, and we're sorry about the mess. Yesterday, it was spread all across the floor, but we thought we'll tone it down this morning. So, oh gosh. Right. I've totally forgotten what I was going to say. Okay, so I'll start again. Sorry about the mess. But we are here with a very important question. Could synthetic biology transform all this rubbish into these new plastic objects? Now, as our name suggests, we find plastics fantastic materials. They're light, they're hard-wearing, they're malleable, and they can be used for so many applications. But, as I'm sure you're all aware, petrochemical plastics are bad news for the environment. But there is an environmentally friendly alternative, bioplastics, and one in particular, poly-3-hydroxybutyrate, or PHB for short. Now, this is a polyester that's naturally made in certain types of bacteria as an energy store. It can be extracted and processed in very similar ways to petrochemical plastics. And it is ideal for medical applications because it's very biocompatible. And we're particularly excited about being able to 3D print with it. So it's a great, great material. However, it's currently very expensive to make because it's made from plant biomass. So what if we could find an abundant and cheap resource to make this material? Oh, wait. So 700 million tons of rubbish is produced on this planet every single year. And the majority of it goes to landfill sites, or is incinerated, or just simply dumped. So this material is actually wasted. Could we use this as a resource to make PHB? So the accumulation of waste and how it's managed is a really complex issue, so we needed to do some research. So we went to Powerday, this recycling plant in London, to find out more about waste management. So we found out that when effective recycling schemes are in place, a lot of valuable material is extracted from waste, but what you have left is this mishmash of different materials that's just shredded up for incineration, which Ian's got a sample of here. They kindly gave us some rubbish to take home with us from Powerday. So, So as I said, this material is currently wasted, but here is a big pile of it behind our new friend Simon Little, who gave us a lovely tour of Powerday. So SRF, so it stands for solid recovered fuel, is made up of one-third plastic, such as polyurethane, and then two-thirds fiber, wood, and paper. So Powerday alone generates 320,000 tons of this stuff every single year. So that's a lot of material to go for incineration. So what if we could use this as a resource to make PHB? If that's possible, then that would revolutionize bioplastic production. So here is our aim. Here's how we're going to do it. So first of all, we're going to turn trash to treasure in Module 1, which we've named Resourceful Waste. So that's cool. If we can do that, then that's a really, really good achievement. But when we've made our bioplastic object, and when that comes to the end of its lifetime, there's currently no way to recycle PHB, so that itself would become a waste. But as we are committed to responsible innovation, we decided that we also needed to come up with a way to recycle it. So that leads us on to Module 2, the recycling platform, which we've entitled Plastic Fantastic. So here's how we're going to do it. First of all, we're engineering E. coli to break down SRF into chemical products and feedstock. Secondly, we're engineering E. coli to transform this feedstock into the bioplastic. And then thirdly, we're engineering E. coli to continuously reprocess this bioplastic as part of our recycling platform. So here's our overview in a bit more detail. So Module 1, resourceful waste. We envisage this to work in bioreactors because we would want to scale this up to an industrial scale. So the SRF goes in. Our bugs would break down the polyurethane part of this waste, and we can extract platform chemicals such as ethylene glycol, which would be filtered off and then sold into industry for manufacturing into other things. And then this would leave the wood, fibers, and paper portion, which could then be put into the bioplastic production, the PHB production. So that's module 1. And then once, as I said, once our bioplastic product has come to the end of its life, we need a way to recycle it. So that leads us onto the recycling platform, where it's broken, the polymers are broken down into the monomers and then rebuilt again, extracted, processed, you have a new bioplastic object. Okay, so I'm now going to hand it over to Ian, who's going to talk about how we designed, modeled, and tested module 1.
Okay, thank you, Gemma. So, in module 1, we produce the commodity ethylene glycol. And we also produce PHB, polyhydroxybutyrate, the bioplastic. To produce ethylene glycol, we need to break down the polyurethane. Now, I should just start by telling you that in our project, we've used the engineering design lifecycle. And this is shown stretched out across the bottom of the screen here. The M1 signifies that we're in module 1. The specifications sets out the requirements of our system. We then design how we're going to achieve these. We model the system to test its behavior, and we use these results to directly affect our wet lab work. Okay. So, on to the first specification. We need our bacteria to be able to grow on this waste without any toxic effects. We're going to use our, first of all, we're going to sterilize the waste before we put it into a closed system. In the future, you can do this by using UV light. So we showed that we used MG1655 as our chassis, as it's able to withstand high levels of ethylene glycol, our commodity product. We showed that it's able to grow just as well on waste-conditioned media as it does do on normal LB. We also showed that it can be used as a sole carbon source by the bacteria for at least up to 6 days, and probably much longer. The second specification we have is that we now need our bacteria to be able to tolerate high levels of our product, ethylene glycol. They can do this. We've tested them, and they can grow well at up to 200 millimolar. We've shown the OD here taken at 6 hours, as it was an easier comparison for us to make with our other results, but we actually carried out the assay for much longer. And literature reports have also shown that they can survive up to 2 moles of ethylene glycol. Now, this is actually a huge concentration, and so we're confident that our cells can survive the levels they're about to produce. So, how we actually do it. We break down the polyurethane using polyurethane esterases. These are called PUR esterases. And this is how they'll work in our system. The esterases will be secreted using a Pelbry secretion tag. And then they will then extracellularly break down the polyurethane, releasing ethylene glycol. The remaining materials will then be used for PHB production in the next part of our module. And these are the designs of our 5 biobricks. We tested SCS2. It was expressed in an active form, shown by it cleaving the substrate here, causing this color change. The color change is caused by the buildup of this yellow 4-nitrophenol. So, onto the second part of our module. This is the production of PHB. So the first thing we actually need to do is to produce PHB. We use the fast CAB operon, biobricked by Tokyo Tech in 2012 to do this. This consists of 3 enzymes which transform the central metabolite acetyl-CoA into polyhydroxybutyrate. When we transformed our cells with this biobrick, they produce PHB, shown by the increased staining here. Okay, what we're about to show you now is quite exciting. This is actually modeling-driven design. And what this means is using our models to directly influence what we did in the wet lab. And this is the model we use to investigate PHB production. So, we ran modeling-guided optimization and carried out a sensitivity analysis to see what is the best way to optimize PHB production. The results show that the system is sensitive to Fab, one of the enzymes' concentration. This is shown by the distance from the x-axis here. We then modeled what happened to PHB production when you increase the level of the concentration of Fab. The results speak for themselves. Increase the concentration of Fab and you increase the amount of PHB production. So, you can do this in vivo by changing to a stronger promoter. This will increase the amount of enzyme present. We modeled these 4 constitutive Anderson promoters here and found that J23104 produces the most PHB. We then created 2 new biobricks from this. One, a hybrid promoter here, shown with J23104 and the native promoter from the original operon, and the second, just J23104. The results were quite spectacular, as you can see for yourself. Both of this is plastic here, we extracted ourselves, and both of these are produced in exactly the same conditions. The top one shows the amount produced by the native operon, and the bottom one, the amount produced by the hybrid operon. We use the industry standard for production efficiency to measure this, and this is the percentage of PHB per dry mass of the cells. We then showed that this had increased this percentage from 5 to almost 60%. This is a massive increase, and it's far higher than any other level that another IGM team has achieved. It's also getting close to industrially relevant levels. So, we've produced a PHB, and we've maximized its production, which is very important in industry. The next thing we need to do is actually produce this plastic from the waste, and we've done that. We extracted this plastic here from bacteria grown on the waste as a sole carbon source. When this was broken down, it was shown that it released 3HB using an assay that Gemma will tell you about in a minute. Okay, so we've achieved a lot of remarkable things in the first module. I'll just take you through them. We've proved that our bacteria can use this waste as a resource. We've shown that our bacteria can withstand the high levels of our product, ethylene glycol. We've not only produced a PHB, we've produced it at industrially relevant levels, and we've also shown that we can actually produce this PHB from the waste. I'll just package you back to Gemma now, who's looking very excited to get on with the next part.
So we've actually done it. We've actually made bioplastic from waste. Whoa! Woo! Awesome. Okay. So, as I said before, because we're committed to responsible innovation, we now need to be able to recycle our bioplastic. So that brings me on to plastic fantastic. All right, so here's how it works. So we need a depolymerase to be secreted from our cells to break down the polymer into the monomer. So you can see here, PHB broken down into 3HB by this Fas1 PHB depolymerase. Okay, so then it's taken up by the cell again, the 3HB, and then with this system, it's built up into the bioplastic. So we've got a BDH2 dehydrogenase, which converts 3HB to acetoacetate, and then using a bit of the endogenous ATO system and our new and improved fast CAB operon, we build it up back into the bioplastic. All right, so here's our specifications. So before we get on to the actual breaking it down, we need to make sure that our bugs can survive in these conditions. They need to not suffer from any toxic effects that the 3HB could have on them. So we've shown this with a growth assay, and as you can see, up until 10,000 micromolar, they grow just fine. Great, so now does the enzyme work? Does it do what we want it to do? So here is our biobrick for that, again with a PEL-B secretion tag. First of all, we showed that it's active. Yes! So with a colorimetric assay, and you can see on the graph as well. But does it break down what we want it to? Does it break down the PHB? It most certainly does. Woo! So you can see after 3 days, there's a distinct clear zone around, oh, sorry, I forgot to mention. So these are PHB emulsion plates. And we cut wells out and then added a cell lysate with the enzyme and also a control. And you can see there's a distinct clear zone around where the enzyme was. So that's really good news for us. But as we've mentioned, we want to scale this up to an industrial scale. So we need to be able to quantify it so we have an idea of how much 3HB is being produced. So we use this kit, which Ian mentioned earlier, that there's a color change to a vivid yellow when the 3HB is produced, and you can compare it to a concentration of 3HB standard. So at the top here, there was no enzyme added, so it's colorless, and then the enzyme was added here, 3HB was produced, and it's turned yellow. So from this, we've estimated that we'd only need about 0.1 grams of the Fas1 PHB depolymerase to break down a kilogram of PHB. So not only is it working on what we want it to, it's actually working really well, so that's cool. All right, so now we've broken down our PHB into the 3HB. We want to build it up again into the PHB to finish off the recycling platform. So for this, we're going to need to take up the 3HB from into the cell. So first of all, we wanted to see if it could actually grow as it's using it as its sole carbon source. So we did another grow assay and found out that it goes reasonably well as a sole carbon source. So now, how are we going to complete it? What enzymes do we need? So, as I mentioned before, we've got our BDH2 dehydrogenase, and also we have a permease to increase the uptake of 3HB into the cell. Now, because we're relying on a little bit of the endogenous metabolic pathway, we needed to find out a bit more how our synthetic pathway is going to interact with this metabolic pathway. So this was another job for our modelers who used this very detailed metabolic model to give us some answers. So we know that we can make PHB from glucose, but we want to make it from 3HB. So we asked our modelers, can we do this? They said, when it's optimized, yes, we can. So we really need this permease to increase the uptake of 3HB into the cell to push the reaction forward. But unfortunately, we're still waiting for this part to be constructed, so you have to stay tuned for this. And now we have 3 extra weeks to work on it. So here is a roundup of Module 2. So, yep, 3HB is not toxic to the cells. Yes, the depolymerase works. Yes, they can use 3HB as a sole carbon source, and stay tuned for the last one. Right, I'm going to pass back to Ian, who's going to wrap up now.
Okay, thanks, Gemma. And the clicker. So, not only have we done that, we've also created a new art form. We call it Bio Nouveau. All of these masterpieces were recreated using our own, more colourful biobricks, which created the pigments shown along the bottom here. We use these to communicate synthetic biology to a wider audience. And people went wild for this. This one down here is a request from Calgary iGEM team in Canada. Okay. We then challenge people's perceptions of waste by directly coupling a future vision of what our system could look like to a 3D printer to show the direct connection between waste and resource. We then challenge people with the question, what would you print? We're back at our home campus of Imperial College. We also raised awareness of the waste issues which affect us personally. And you might have seen some of our posters around the place yesterday. Okay, so what we've done in this project is completed the loop on two things. First of all, the waste, and secondly, the bioplastic, moving us closer to a circular economy. And okay, we've successfully turned trash into treasure. Thank you very much. Any questions?